back. Welcome. So I'm now going to tell you about Genoscopy. I mentioned that I founded this company as a medical student, and we feel very fortunate to be a part of this event. So I'll let you know about kind of what we're doing in terms of the numerous applications of stool-derived eukaryotic RNA to assess gastrointestinal disease. So Genoscopy's platform is pretty cool. It was technology that was developed at WashU that we pulled the technology out and founded the company. We basically ship collection kits to patients across the United States. We have them deposit a stool sample based on our IFU and ship the stool sample back to our centralized laboratory. In the laboratory, we isolate the eukaryotic cells, preferentially to the bacterial noise that we describe, and then we perform queries to extract, isolate, purify, and quantify the RNA based on our clinical questions that we're asking. Using this platform, we can successfully isolate eukaryotic RNA in over 95% of samples that are shipped in the mail at ambient temperature for up to 3 days. The analysis platform that we utilize is very robust. The types of biomarkers we've been able to pull out of stool samples includes RNA expression, expressed RNA variants, and protein concentrations that are correlated with disease. We have been able to evaluate enterocytes from the colon. We've been able to evaluate T cells, B cells, and now in the wave of COVID, we've actually been looking at bacterial cells and viral capsids to better understand disease. And our platform is really quantification agnostic. So we've pulled and evaluated the RNA out of these cells and then put them on a numerous different platforms, including next generation sequencing, digital PCR, qPCR, molecular barcoding, and microarray. We've also developed a really unique way to obtain patients. We enroll completely online. It's a very decentralized approach. We have a centralized nurse call center that does informed consents and obtains medical data release forms. And then we directly drop ship collection kits to patients anywhere in the United States. We use FedEx to have them brought back to our laboratory, and then we use a central lab to evaluate the RNA. In total, over the last year, we've had over 270,000 individuals engage with our online platform. We have 60 nurses at our call center to do the informed consent and medical data release forms. And we have really high engagement from a diverse group of individuals, not just people who are coming to the hospitals, very health literate, aware, have insurance, things like that. We're getting a really high level of diversity and inclusion in our clinical trials. And we have about 85% compliance of returning the kit to our laboratory using this process. We've used this platform to develop our lead assay, which is a non-invasive test to assess average risk individuals for colorectal cancer and advanced adenoma. In preclinical trials, we've demonstrated a 95% sensitivity for colorectal cancer. We've demonstrated 100% sensitivity for individuals that have advanced adenomas with high-grade dysplasia and 62% sensitivity for all advanced adenomas. This is compared to a screening colonoscopy. And I'm sure many of you are familiar with Cologuard, which is the non-invasive method for average-risk individuals to screen for colorectal cancer. So relative to that pivotal study that they conducted in 2014, we demonstrate about a 50% improvement in adenoma sensitivity. The clinical validation study, which is a 14,000 patient prospective clinical trial, was launched in 2021 of last year. We've actually completed enrollment of 14,000 patients and anticipate submitting this as a PMA to the FDA this fall. Finally, while most of our efforts have been devoted towards colorectal cancer screening, we have numerous applications within the colorectal cancer space in terms of predicting therapeutic response. We've also advanced into inflammatory conditions like IBD and IBS, and we have a companion diagnostic for FAP. So I'm very happy to show the progress we've made over the last couple of years, and I'd be happy to engage with anyone who'd be interested in our company and working with us in the future. Thank you.